So Rare Diseases Day is an international event that's held on the last day of February each year to really raise awareness for rare diseases and the impact that they have. So the definition of rare disease is complex and it varies in different places around the world. We tend to go with the European definition, which is that a rare disease is one that affects less than or fewer than 1 in 2,000 people in the community.
So at the moment we are focusing on Ataxia Telangiectasia, which we shortly call AT for obvious reasons. This disease occurs in about 1 to 40,000 to 1 in 100,000 births, so it's pretty rare. But when you get it, it's pretty bad. So these children that develop AT have a high prevalence of cancer. So many of them get blood cancers, but that's actually not what kills them. What kills them is the degeneration of their hindbrain, the part of the brain that controls your breathing, your movements, your swallowing. And that's the main problem that we're trying to solve.
So we've been really fortunate here to study a number of different rare diseases. We've studied a lot of diseases that affect the growth and development of the skeleton, what we call skeletal dysplasias. So everybody who has dwarfism has a skeletal dysplasia, but not everybody with skeletal dysplasia has a dwarfism. And so we're trying to find the genes that cause these rare conditions. We've studied conditions called short rib polydactyly genes, and we've found about 4 of those over the last few years. And this is incredibly helpful because it fills in the gap in the pathway. So we identify a gene that causes a condition, and that tells us a little bit more about the cycle of how this gene interacts with other genes and how these proteins are affected. This is important because from a family's point of view, it's an explanation. It tells them why their child has this condition, sometimes confirms what condition their child has, tells them what the risk is of having another child with the same condition, and also allows them to reach out to other people whose children are similarly affected as well.
I work on a very rare group of disorders called Leukodystrophies. And Leukodystrophies affect the white matter of the brain. So you can think the brain contains a whole lot of neurons that carry all of the messages around the brain. The white matter of the brain is the insulative layers that separate those neurons and make sure the messages get to the right places. In leukodystrophies, that white matter either never develops properly or it's degraded over time, and it means that the signals in the brain no longer can get to where they need to go. And it can have a very devastating effect on the children that it affects. So we're focused on identifying the genes which are, when mutated, cause leukodystrophies. And so we've been working with an international team of clinicians to collect patients who have leukodystrophies and sequence their DNA and try and identify the genes that have the mutations. And so far, as part of that team, we've identified 6 new genes that, when mutated, can cause leukodystrophies. And we're also trying to use the approaches that we're using in the lab and get them applied in a normal clinical setting, which will allow very rapid diagnosis of children with leukodystrophies.
I'm interested in understanding how the heart forms and how it functions. So we know that the genes in our body, so in our DNA, tell the heart every time to form in exactly the same shape and exactly the same way so that it functions to keep us alive. We also know that if we have mutations in these genes, that we have defects in the way our heart forms and functions. Here at the IMB, we've performed a large screen for genes involved in heart development. What we've done is we've taken animals, and each of those individual groups of animals have different genetic mutations in them. And then we've looked in those groups to see if they have a heart defect. When they have a heart defect, we know that gene in particular is causing a heart defect, and then we can compare that with patients and see if they also have defects in those genes, if they have mutations causing their rare disease. So we don't have the technology currently to fix these types of genetic disorders, but on building this fundamental knowledge and this basic information about how these defects arise, we hope that this will help us later to develop things such as drugs or alternate genetic technologies that might help to fix these sorts of diseases in the future.
I work on a group of diseases called disorders of sex development. Some people refer to these as intersex or variations in sex development, and as the name implies, this refers to when children are born with sex organs that don't reflect the typical development of these organs. This is a broad spectrum of disorders. Some of them are relatively common, some of them are much less common, but as you know, with rare diseases, the total number of children affected is actually quite large. One of the major questions that arises when a child is born with one of these conditions is, how did it arise? Depending on the cultural background of the family, there might be a belief that it's just something that's occurred through bad luck or an act of God or something. So our research provides solid scientific explanations as to what went wrong at the genetic level. This helps us to provide better diagnostics for these conditions, clear explanations to clinicians as to what the outlook might be for the further development of the child, and that of course guides the clinical management of these conditions.
So I've been working on rare diseases for many years. I did my PhD in London many years ago on cystic fibrosis, and it was at a time when we didn't have the sequencing technologies that we have today. And I think in the last sort of 5 to 10 years I've become more interested in rare diseases again and looking at how we can use these technologies to help patients in diagnosis and treatment.